be discussing about the juxtaglomerular apparatus. In the last class, we have studied about the structure of the nephron. I hope all of you are aware that the nephron has two major components. That is, we have the renal corpuscle. The renal corpuscle is there as well as the tubular component. The corpuscle has the Bowman's capsule as well as the glomerulus, and the parts of the tubule are the proximal portion, which has a proximal convoluted tubule and the straight tubule, the loop of Henle, which is made up of the thin descending limb, the thin ascending limb and the thick ascending limb. Then we have the distal convoluted tubule, the connecting tubule, which then tapers off as the collecting duct. So, in today's class, we will be concentrating on this specialized structure known as the juxtaglomerular apparatus. Now, here there is this structure is where the glomerulus comes in contact with the thick ascending limb of loop of Henle. It is a specialized structure. It has both the vascular as well as the tubular components. So, here in the juxtaglomerular apparatus as such, we have three major parts. First one is the juxtaglomerular cells, which are found in the region of the afferent arteriole. They are the modified smooth muscle cells of the afferent arteriole. Then we have the macula densa, which is a portion of the thick ascending limb of loop of Henle, and we also have certain supporting cells in this region known as the Lassus cells. So, coming to each one, first about the macula densa. So, it is the thick ascending limb of loop of Henle. There are certain modifications in the cells over there. They form the macula densa. Then the juxtaglomerular cells are actually the modifications of the muscle cells, smooth muscle cells of the afferent arteriole, and we also have the Lassus cells or the supporting cells. Then the main innervation of the juxtaglomerular apparatus is sympathetic. Then coming to the juxtaglomerular cells, they are actually the specialized myoepithelial cells, which is located in the tunica media of the afferent arteriole. So, the major function is they synthesize, store and release renin. These cells, they have abundant endoplasmic reticulum, Golgi apparatus. Apart from that, this renin that is secreted from the JG cells is stored in the form of granules. So, as and when it is required, these cells, they respond to pressure changes in the afferent arteriole, and it is stimulated by decrease in the blood volume, hypovolemia. Then coming to the macula densa. Macula densa is actually the modified epithelial cells in the thick ascending limb of the loop of Henle. These cells actually act as the chemoreceptors, and they get stimulated by changes in the sodium chloride concentration, and this can also influence the renin release. Then coming to the Lassus cells, now they are the supporting cells. They are the interstitial cells of the JG apparatus, and these cells are in close contact with both the juxtaglomerular cells as well as the macula densa, and they relay signals between the macula densa and the juxtaglomerular cells. Now, coming to the functions of the JG apparatus, two major functions. First one is it helps in the regulation of blood pressure, that is through the renin-angiotensin aldosterone axis, as well as the second function is it also helps in maintenance, regulation of the renal blood flow and the glomerular filtration rate via the tubuloglomerular feedback. So, coming to the regulation of blood pressure via the renin-angiotensin aldosterone axis. Suppose there is a condition of hypovolemia or hypotension, as in when the BP is decreased or if the blood volume is very less. What happens is the baroreceptors that are present in the kidney, they are not stimulated. So, in such situations, there is sympathetic stimulation which occurs, and this sympathetic stimulation leads to the release of renin from the JG cells. And once this renin is released, it helps in the conversion of angiotensinogen to angiotensin 1. Then another enzyme, ACE, angiotensin converting enzyme, which is produced from the lungs, helps in the conversion of angiotensin 1 to angiotensin 2. Now, angiotensin 2 brings about all the actions, that is, it increases the blood volume and the blood pressure by powerful vasoconstriction. Angiotensin 2 is a very powerful vasoconstrictor. It can also, this angiotensin 2 can also act directly on the kidney to cause sodium reabsorption, as well as it also causes the release of aldosterone to cause sodium reabsorption. Then coming to the regulation of renal blood flow and the glomerular filtration rate via the tubuloglomerular feedback, what happens is in cases where there is increase in the blood pressure, the renal blood flow and the glomerular filtration rate, what happens is there is increased delivery. The tubular flow will contain excess quantities of sodium. So, what happens is the macular densa, the thick ascending limb of loop of Henle also receives a lot of sodium. In such cases, what happens is there are certain changes take place which ultimately cause afferent arteriolar constriction, which can bring about a decrease in the GFR. So, what actually happens is, please take a look at this diagram. Here we can see the macula densa. This is the modification of the thick ascending limb of loop of Henle. Then this is a tubular lumen. Then we have the arteriole here, afferent arteriole here. What happens is if there is increase in the GFR, renal blood flow, what happens is the tubular flow to the tubular flow will also increase. In such a condition, what will happen is there is increase in the sodium and chloride concentration along the tubules. So, this sodium and chloride, it is picked up by the sodium potassium 2 chloride transporter, and hence what happens is because the increased concentration of sodium, the sodium potassium ATPase activity is also very high. More amount of ATP and adenosine are formed. This adenosine will act on the A1 receptors, and this A1 receptors will cause the calcium influx into the arteriole. Once calcium enters into the arteriole, there is vasoconstriction. When there is vasoconstriction, there is decrease in the GFR and the renal blood flow. This is the regulation of the renal blood flow and the GFR through the tubuloglomerular feedback. So, now summarizing the whole thing, the juxtaglomerular apparatus, it is mainly made up of three major types of cells. The first one is the juxtaglomerular cells, which are nothing but the modifications of the smooth muscle cells in the tunica media of the afferent arteriole. They mainly contain, these cells, they contain granules which secrete renin, and this renin, by its action, it is able to increase the levels of angiotensin 2, and this angiotensin 2 brings about vasoconstriction and other changes. It also causes aldosterone secretion, increases the sodium reabsorption. Then after that we have the thick ascending limb of loop of Henle. This portion here, the cells, you can see they are much taller and narrow cells with the nuclei closely packed together. These cells, they are the macula densa. They sense the sodium chloride concentration. They are actually the chemoreceptors. They sense the concentration of sodium and chloride, and appropriately they bring about changes in the renal blood flow and the GFR. Then we have the Lassus cells, which are nothing but the supporting cells which are found between the macula densa as well as the juxtaglomerular cells. So, the components once again, they are the juxtaglomerular cells, the macula densa and the Lassus cells or the extraglomerular mesangial cell, and the functions are juxtaglomerular cells, it contains granules. Granules release renin as and when required, and through the renin angiotensin aldosterone system, it has a role in the regulation of blood pressure. There is a macula densa is involved in the tubuloglomerular feedback mechanism and thus controls the GFR, and hence there is a role in the juxtaglomerular apparatus, has a role in the control of the renal blood flow. Now, this JG apparatus as such, it can be asked as a short note. Sometimes one word answers MCQs can be asked. So, one such example is juxtaglomerular apparatus contains all cells except. So, Lassus cells, they are the supporting cells. Macula densa is a modification of the thick ascending limb of loop of Henle. It is a part of the JG apparatus. The juxtaglomerular cells are modifications of the afferent arteriole. The glomerular mesangial cell, option D, is not part of the JG apparatus. Then Lassus cells, they are located at the juxtaglomerular apparatus. Now, this is a straightforward question. It is not present in the proximal tubule, distal tubule or the loop of Henle. So, the answer is juxtaglomerular apparatus. Okay. Thank you.